one class that helped you.
All right, let's go ahead and start. Okay, so first things. Remember that a week from today is the second exam, and that at 2 o'clock on Sunday, I'll have another review session. Anybody wants to show up. So this time it is in the Anderson 112, also known as the Nelson Auditorium. It's on the first floor, down about there. Okay? Second of all, I just thought you might be interested in this. It just came out last week. The metabolic regulation of female puberty via the hypothalamic AMPK kisspeptin signaling. So we talked about how kisspeptin is regulating the LH surge during ovulation. So this is a paper that just came out in Proceedings of the National Academy of Sciences. It's usually a pretty big deal. And what they're showing in this paper is that kisspeptin also plays a huge role in female puberty, human puberty. So they're still working out how it works, but if they knock it out in the rats, you didn't see puberty occurring at all. So it looks like this hormone, this released neurotransmitter in the brain, is having a big effect not just on ovulation but also on puberty. So now I have to rewrite my lectures for next year talking about kisspeptin. Okay, so back to stress. So we talked about these two different responses. The fast response, which is the fight or flight, we talked about that last week. And then we're now talking about these slow responses with the cortisol, corticosterone. Which of these, remember, which of these versions of the glucocorticoids is species specific and doesn't seem to make any difference? So we were talking about these two figures. So this idea of very different kinds of novelty. And we talked about the paratroopers and the fact that you can have different contexts can have a great difference in whether or not you have a stress response at all. There are other kinds of contexts that we also has been shown to have a dramatic impact on whether or not you have a stress response. One of which is this, advanced warning. Yeah.
I'm not sure we started this week here.
Didn't I talk about this? Yeah. Okay. So this is the one of the paratroopers being taken up on the towers, being thrown off. We can talk about it again if we need to. Okay. Advanced warning. So what these researchers did is they took two different cages, and they had a rat in each cage, and they would shock the rats. And guess what happens when you shock a rat? Its glucocorticoid levels skyrocket. They don't like that. So here is when they were not shocked at all. So this is the control. And when you shock them, this is the responses you get. This is not surprising, right? What they did, though, is they took on one of the other cages. So one cage just got shocked, and the other one, they simply turned a light on a second or two before the shock. And so the rats learned that as soon as that light turned on, they were going to get shocked. Notice that both rats got exactly the same shock for exactly the same duration. And yet, one of them, who got the warning, had a much lower response. So it's not, when we were talking about what causes stress, it isn't just an event, a stressor, that can elicit a response. It's also dramatically important as the context of that stressor. And simply knowing that it's coming, the rat could steel itself, and it has much less of a response. Okay? A similar experiment is not even just the advanced warning, but also the control. So what in a similar experiment, they'd have two rats. Both of them got the same amount of shock, but one of them could run over to a lever and turn it off for both of them. And so they both received exactly the same shock, but the one that had the control over it was able to turn it off. And so both this advanced warning and the ability to turn it off, to have control over it, also has a major effect. So both novelty and control are things that can dramatically impact stressful events. And this is one of my favorite experiments ever in terms of thinking about stress. Let me explain to you what's going on here. So what they did was they trained a rat to press a lever to get food. So it was going to get a reward. So every time it pressed the lever, it's not every time, but every 20 presses, it got a reward. So it just said there, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, a reward. And then it could do it again. And that's what the FR is, fixed rate. So if they had a fixed rate 20, they were getting a food reward every 20 presses. This is their glucocorticoid levels beforehand. This is after the training. Obviously, no effect of this. If they go from fixed rate 20 to a CRF, which in this case means constant reward, okay, constant rate. So instead of every 20 presses, now they get a reward every time they press the lever. And guess what? Their stress levels go down. Not surprising, right? Interestingly, if you go from a fixed rate 20 to a fixed rate 10, so now you get twice the amount of rewards for the same amount of work, there was no effect. But here's the interesting one. Fixed rate of 20 to extinction, okay? Which means that now they never get a reward ever again for pressing this lever. Guess what? Rats don't like that. You wouldn't like that, right? You get used to getting a reward, and yet now you're never going to get a reward again. And so there is a large stress response. But this is the one that I find really cool. They went from a fixed rate 20 to a fixed rate 40. So in other words, all they did was halve the amount of reward that they got. And yet, they still have a massive increase in their stress response that's roughly the equivalent as if they never saw a reward ever again. Notice, this is not shock. We're not punishing this rat. All we've said is that you're getting a smaller reward than you used to. Anybody think that this has anything to do with humans? Because anybody who's had a toddler knows that you can give them a piece of cake, but if somebody else got a bigger piece of cake, they're still unhappy. Right? So this is one of the things. I could guarantee you that I could hit you over the head right now, and you would see a decrease in your stress response. How can I do that? Yeah? Well, you don't know that I've hit anybody else harder. It's even easier than that. Yeah? But they do tend to see big changes. So a lot of people who are well, so right now, I'm about to come up and hit you on the head. How could I possibly claim that you're going to have a lower stress response? You're going to actually be happy. Yeah?
You can tell them it's not going to be as bad as you think.
You really think that getting hit on the head is going to be better than you think?
Well, no, I'm saying like it's not going to hurt. It's like when you're getting a shot, they tell you, oh, it's not going to hurt. It's kind of like that.
Yeah, but this is going to hurt. I want to hit you hard enough so it hurts. Yeah?
You said like every sixth person gets stabbed instead?
No, it's still going to hurt.
I mean, but that would be relieving that you got hit and stabbed.
Yeah, it's even easier than that.
Okay.
Yeah?
You tell us your next class you're going to hit them twice.
Why next class?
Because it.
So the other organization, by the way, who was focused on a lot of studying. You're almost there, but you got it reversed. There you go. Maybe not even the previous class, but just every class I come in and I hit everybody on the head 5 or 6 times during class, right? This is going to get really, really annoying. You're going to have big stress responses coming to my class. But then I come up and I only hit you once. Guess what? Things have improved. Right? Things have gotten better. And that's the key to thinking about stress responses, is that there is this discrimination determination thing that's in our brain that is looking at what is happening before and comparing it to what's happening now. And it's that comparison that is going to either increase or decrease your stress response. It's not an objective thing, it is entirely subjective. So if you are being used to hit over the head every 2 or 3 minutes, and now all of a sudden I only do it once, things have improved. You're no longer, not only do you not have a stress response, but your actual stress levels go down. So think about the way that works. Just simply, if your things improve, even though you're still getting a noxious stimulus applied, you think things are getting better and you don't have a stress response. And if things are worse, even if you're continuing to get a reward, like these rats, you're still going to have a stress response. It's entirely in that subjectiveness. Hopefully you're starting to understand why studying stress has been so difficult. Okay, a couple more things. Facilitation. So there are ways in which the body will ramp up stress responses. So this is kind of, it's actually a simple design, but for some reason they made it really difficult when they put this into this figure. So here's the key here is that this is the treatment at 9 in the morning on these rats. And this is the treatment at noon, 3 hours later. So, and this is the level of plasma corticosterone in these rats, the stress response. So if they do nothing at 9 a.m. and they do nothing at noon, and then take a blood sample, this is what you get. So there's your controls. If you then inject saline at 9, so you grab the rat, and all you do is injecting saline, and then you come back 2-3 hours later, and then you give an injection plus 15 minutes of a restraint stress, of a stressor, which they don't like. They have this response. This is not just an increase in corticosterone, because if you inject corticosterone and then do a 15 minutes of stress, it's much lower. So this is actually the brain detecting and realizing that things are bad. But if you can get restraint earlier, and then again restraint later, 3 hours later, not only do you have the same response, but you have a higher response. And so this is what has been called facilitation. By layering different stressors on top of each other, you can continue to get higher and higher responses. This is one of my experiments. I did a lot of this when I was in graduate school to try to understand how glucocorticoids are being released. And this is what people have called frustration. And then what we do with this is that you provide a, normally when you have animals in captivity, you provide them constant amounts of food and water, everything they want. In this case, what we did is we restricted their food and their water so that they only got an hour every day in which they got water or food, and then it was taken away. So basically a 23-hour fast every single day. And so that they start to anticipate that this reward is going to be coming. It's really not a reward, but they're actually going to get their food and water. And when they get given that, what you see is that it immediately, all of their stress levels go down. So this is when they're given both their food and the water. When they're not given either of them, what we see is they get frustrated, so that all of a sudden their reward was not given. And so you see this massive increase of their corticosterone levels. That's not surprising. What is really interesting, though, is that these levels here. You're talking about the difference in energy. What they get given is an empty water bottle and a food dish with no food in it. And so simply getting that reward, we immediately see this massive decrease in corticosterone that then shoots up. I mean, think about it. So there is a regulation that's taking place at the level of the adrenal gland that is regulating whether or not glucocorticoids are released, simply depending on whether they're expecting the reward as well. Why? Yeah. Okay? Why do we not taste it? And here's a study from the early 70s called SIP, or schedule-induced polydipsia. So one of the things that people discovered quite early is one of the ways in which animals can sort of, and this is people too, can reduce their stress is to have a displacement activity. So in these rats, what they do, if they have a water bottle and they have a shock underneath their cage and they get shocked, what the immediate thing they'll do is they'll run over and start to drink. And it's a way of displacing that kind of fear and stress that they've gotten from just being shocked. So what they showed is that if you increase the time, the interval between the shocks, that they get so anxious that they're about to get shocked that they start to drink more and more and more water. And to the point where at about, this is 180 seconds, so about every 3 minutes, they are now drinking so much water that they can overdrink and actually die. They can literally drink themselves to death with water. That anxiety about being shocked, on average about every 3 minutes, just is so powerful that they will drink themselves to death. And then if you have more than 103 minutes, then all their drinking goes down. Now they have enough time to rest. that it's not nearly as stressful.
And so they come up and they
So,
taste, even
two
though
things
they
about
taste
this
good,
study. First of all, this was going to be, in the early 70s, the model for understanding drug addiction.
eating and dying and
Because as you can see, these individual rats it was so potent for them to want to reduce that stress that they were drinking water to the point where they were dying. And so this was, people proposed, okay, this is how we're going to understand the biology of addiction. Turned out
enough fat.
to be not useful at all for understanding addiction, and the reason was particularly interesting. It turns out to be this response is almost entirely an artifact of having rats that are domesticated. So about 2 or 3 years after this came out, people went and repeated this experiment in wild rats, freshly caught captive rats. It turned out no effect at all. The second interesting thing about this, however, is what is the reason I keep showing it, because I think it's really cool. 3 minutes. That kind of pressure that's on you for having a stressor approximately every 3 minutes is so potent that these rats can drink themselves to death, but it's also the same if you flip it in terms of reward. Turns out 3 minutes with a variable rate, but on average a reward every 3 minutes is exactly the period that they use in slot machines. Okay? And they didn't come up with that from this experiment, but it's clear that it's the same basic phenomenon. So it's so impactful for us that people can't stop feeding coins into
One
slot
last thing
machines.
to think about in terms of
So think about that next when you ever go to Vegas or Atlantic City and you walk past all those slots. They are designed to do this to us.
So there are 2 different types of diabetes.
Okay?
There is the insulin
Finally.
dependent.
Social context has a huge impact.
And it's not.
So this is a series of experiments that were done on wild baboons. But a lot of people have used these data to explain what's happening with humans as well. So let me explain to you what happens in a baboon troop, especially baboon males. Somebody goes out and they either find or they kill something. And so here's your male who's just killed this antelope and is sitting there eating, and he is not the alpha. Here's the alpha. So the alpha comes over, and this lower ranking male baboon abandons his food because the alpha is going to come and is going to take it from him. So it turns out that this lower ranking male does have a stress response when this occurs. But there's interesting things about when a stress response occurs and how large that stress response actually is. So for example, this is a threatening situation. The alpha comes up and tries to displace you. But what if the alpha male is just walking past you and is not being interested? If you can tell the difference between a threatening and a neutral interaction, you have a lower stress response than if you can't tell. So if every time the alpha walks past you and you get anxious, you're going to have lots and lots of stress responses. If you actually initiate a fight with a threatening rival, so even though that alpha male comes up and starts to harass you, if you turn around and fight, you actually have a lower stress response than if you just took it. But are you likely to win that fight? So the ones that are most likely to win that fight have a lower stress response than those that are not as likely to win the fight. And one of my favorites, the differentiation between winning and losing a fight. If you could tell that you won or lost the fight, even if you lost the fight, if you could tell that you lost it, you had a lower response than if you couldn't figure out whether you won or lost the fight at all. You have a larger stress response. And finally, are you likely to displace the aggression after losing the fight? So generally what happens, the alpha male comes up and gets into a fight with a beta male. The beta male loses. What does the beta male do? It walks over to the next male and beats him up. Displaces his aggression. The stress response goes down. So what does the next male do? Walks up to the next one in the hierarchy and beats him up. And then he goes and beats the next male up. And so this cascades all the way down the troop. And so finally the lowest ranking male has just gotten made up. What do you think he does? Goes and beats up a female. And then then it goes down the female hierarchy. So what you see is that this real important idea of displacing this aggression, displacing the fact that you've lost, actually has a major impact on whether or not you're going to have a stress response. Okay, yeah. Yep, but just starting one that you know you're going to win, because it's a lower rank. Oh yeah. But you don't want to lose. If you lose, then you tell, well, did you know you were going to lose? See? So it all feeds back. So a lot of this is just cognitive.
What are you going to look for in the blood?
All right. So now let's talk about what glucocorticoids actually do. This is whether or not they get released or not. What do they actually do? So now we're going to come back to this figure. So one of the most important things that glucocorticoids do is they increase glucose. That's why one of the reasons they were named glucocorticoids. Why? Because they impact glucose levels. This is how they do it.
That's what you have basically an outside tube running from your mouth to your
So you have a stressor.
So all of that stuff is going right through.
Remember, the first and initial response is to release epinephrine. That takes place in under seconds. And epinephrine, if you remember when we talked about the catecholamines, one of the things, and glucose regulation, one of the primary functions of the catecholamines, epinephrine, is to go and inhibit, shut down insulin so that insulin doesn't produce glycogen, and then to break down glycogen. That's this gluconeogenesis. So this is glycogen breakdown.
Here's to just put this in context. One of the things you probably all know is that stomach acid, stomach contents are.
So that's our real initial response.
Throw off all that acid.
The glucocorticoids are released a few minutes later,
Is acid secretion?
and they have three major functions on regulating glucose levels.
How do we figure
One of them is to make the epinephrine work longer and better. So remember, I showed you, when the glucocorticoids are coming down through the adrenal cortex, before they get to the rest of the body, they're passing through the adrenal medulla and impacting the production of PNMT. So this is a permissive effect on epinephrine working to stimulate gluconeogenesis by glycogen breakdown. Okay, so glycogenolysis, breakdown of glycogen. The second thing is that glucocorticoids will decrease peripheral usage. So they go to most of the cells in the body and they stimulate the production of a peptide called sequestrin. So again, thinking about how insulin worked. Insulin was one of the major functions of insulin was to put glucose transporters onto the cell surface so that you can get more glucose coming into the cell. What glucocorticoids do is they produce sequestrin, and sequestrin then takes those glucose transporters back off the membrane. And so all the cells have less glucose that they can work with. And by having less glucose go into the cells, what you see is then an increase in blood glucose levels. Why is that important? So the idea that people have had is that you increase blood glucose levels to allow things like muscles and the brain tissues, etc., to have preferential access to that glucose.
out of our intestinal tract?
Make sense?
So we include to the rest of the body. So there is a
Yeah.
lot
So
of
are the transporters those like muscle and
always
cells
simple.
not
And you wouldn't want to get the artery, would you?
That's the hypothesis. It doesn't always work that way. So interesting, I was trying to, my first experiment that I ever did in graduate school. So was looking at exactly this phenomenon. Because what other tissues would really, really want to have high levels of glucose that they have access to? Tumors. So it turns out, after a lot of experiments, that we found out that tumor cells also shut down their ability to listen to glucocorticoids, and they don't create sequestration. So all of a sudden, the body, under stress, has increased the amount of glucose in the bloodstream. All the body is taking up less glucose, except the tumor. The tumor has all access to all that extra glucose, which starts to explain why stress can cause an increase in tumor growth. Yeah.
Is this spike in glucose also what leads to like the metabolic?
So there's created a port
Yep.
into the animal's
Yeah.
stomach.
So the new thing that people are talking about is
Which you could go in and look and see
metabolic syndrome.
digestion. So in this
And a lot of that has to do with the glucocorticoids that are
they make a
creating
port
this.
big enough that they
It has a lot to do with
can
glucose.
break down and grab the digestion
Okay, so
stuff.
sequestration then decreases the peripheral usage. And then we have this long-term effect
And so to try to figure out
where glucocorticoids start stimulating gluconeogenesis. And this is from
what the cells are,
protein
we have a variety of different
breakdown.
approaches. So here are different kinds of approaches that you can use.
So we start to break down our proteins in order to get more glucose into the body. Okay? So we have these really short-term effects that are permissive. We have these activational effects that are with the sequestrin, and then we have these long-term
But you can do
effects.
other things as well.
So if we think about how glucocorticoids work, and this is a good example with glucose, but it's true of a lot of other systems. What we see is there are 4 major classes of glucocorticoid actions. They have permissive actions. So this is like the allowing epinephrine to work better and longer. They have the stimulating actions, like increasing blood glucose levels. They also have suppressive actions, in which you have effects where you shut things down. And this is primarily going to be the immune system, which I'm going to talk about in a second. And they also have preparative actions. And that's where you get this kind of response, these preparative actions. Okay? So why? So in many ways, this is the more interesting effects. And why do I say that? Think about a stress response. You're sitting in the Serengeti, and a lion jumps out to attack you. What's the first thing your body's going to do? What is going to increase? Epinephrine. You're going to need your fight or flight response, right? Now, I showed you glucocorticoids aren't going to be released for about 3 to 5 minutes. Plus, what they really are, in many cases, are transcription regulators, right? So, how long until we start to get an effect from glucocorticoids?
So we're not really going to talk about what's happening up here in the oral cavity.
Maybe 20, 30 minutes? All right. How useful is that glucocorticoids going to be in escaping from that lion? Basically 0, right? So, what are some of the reasons why glucocorticoids are released? And this comes with, I'm going to give you an analogy that'll try to explain how we're thinking now about what really glucocorticoids are doing in most cases. You got a country. Country gets attacked. What's the first thing the country does? What? Yep, how? So mobilizing your troops, right? So mobilize your troops, get them out of the barracks, send them out and fight. That is essentially a fight or flight response. We've already built those things. We're ready to go. Okay? However, how do we know how long this conflict's going to last? What other things would we start doing? Well, we might start instituting rationing for the civilian population, so we get more of that food to our troops.
that's embedded
That is a
inside the
longer-term
liver
thing.
and the liver itself.
Okay? In some ways,
Another part of
that's
this is the
a
pancreas.
stimulating action, but it's more of a preparatory action. We're getting things ready to go. Another thing,
And the
we might,
pancreas is
depending upon how the other country attacked us, we might start building battleships
Now we have to talk about its
because we
pancreas.
think we might need them. How long does it take to build a battleship?
Okay, so these are the major areas.
I don't know how long it would take to build a modern battleship, but in World War II, it took us about 2 years.
In greater detail.
Okay,
This is
so
what
we started building battleships, and they're not going to be able to use them for 2 years.
This then is.
That is the kind of preparative action. And that's a lot of what the glucocorticoids are doing. They're preparing the body not to survive that first response, but to survive your later responses.
Gastric pits.
Okay?
Long areas.
And I'll show you why in a minute.
All of the
All right,
digestive stuff to get down.
so we have to keep those 4 things in mind. So we talked about the increase in blood glucose levels. The other thing, the major impact of glucocorticoids is to inhibit the immune system. So what is it? What do glucocorticoids have? Their effects on the immune system. There's a lot of them, but they all essentially are inhibiting the impact of the immune system. So they inhibit synthesis, release, and efficacy of cytokines. So the cytokines are essentially the hormones of the immune system, and so they're shutting all that down.
And the chief cells are
They inhibit antigen presentation. So this MHC, major histopathology complex expression. These are the things in your lymphocytes that actually allow you to determine self versus non-self and help you fight off an infection. And the glucocorticoids inhibit that presentation. They reduce the activation and proliferation of T cells, B cells, and macrophages. So they really shutting down the major cells involved in the immune system. They lower the circulating levels of lymphocytes. These are one of your primary ways of fighting infection. They reduce lymphocyte chemotaxis. So one of the things that lymphocytes do at the site of inflammation or site of an injury is they actually leave the blood and move into the tissue to fight the infection. Glucocorticoids inhibit that function. This, by the way, reducing inflammation. Many of you have probably taken or used a glucocorticoid. It is one of the most popular hormones and drugs prescribed by doctors, primarily as a cream. So people have used cortisone creams. Cortisone is a synthetic glucocorticoid. And what does it do? You put it on where it's itching, it's inhibiting your inflammation right there at that site. So it's one of the big things it does is inhibit inflammation. The thymus is a major organ for immune function, and this glucocorticoids will atrophy the thymus. And they can even trigger death of immature T and B cells, leading to immunosuppression. So one of the big things you can do is shut down the immune system when you have chronic stress. Yep.
So what if that chronic stress though is in the form of infection? If you're losing yourself on
Yes, you do. So the question is, why is it doing that? And that is, we still don't have a good idea for that. Yeah.
How much energy does it cost to do all of these additional functions of the immune system? Is it?
How much energy does it cost? It's been an area of great debate over the last number of years. The consensus is it doesn't hurt. It's not too energetically expensive. Because one of the things that also happens is that we start to see sickness behavior. And that is actually also regulated indirectly by glucocorticoids. So you get to feel more sick. That means you become less active, and that then saves a lot of the energy that you're using for some of these other effects. Okay? Turns out that it's a little more complicated than this. One of the great experiments is looking at the difference between acute and chronic immunosuppression. So this is an example where what they did is a really neat experiment. What they're looking at here on the y-axis is what's called the thickness of the pinna. So what they did was they take a a injection and inject it right into the lobe of the ear. And it's not an infection. It's oftentimes what they'll use, the bacterial cell walls. So it's not a bacteria. It actually doesn't cause an infection, but it does stimulate the immune system to react. So the immune system reacts. A lot of the T cells, the B cells, a lot of the macrophages, they're all flooding into this area in order to fight this, what they think is an infection, and so you start to get inflammation in this area of the ear. And so you can measure the amount of inflammation when you do that to a rat or to a rabbit or something like that. And what they did was they showed, this is the controls, this is the response, the amount of increase in the size of that earlobe when there's in the controls. When you stress the rats beforehand, you see a much larger response. You've essentially primed the immune system. So in fact, it's exactly the opposite of what I just said. You're getting an increase in the ability to fight the infection, not a decrease. And we know that's regulated by glucocorticoids because from this panel, here's your controls, here's a low level of injection of corticosterone, and here's a higher level of corticosterone. So the corticosterone itself is actually helping mediating this effect, getting an increase in the amount of inflammation. But this figure is the critical one. So here's our controls, and this is chronic levels of stress, chronic amounts of glucocorticoids. And what we see is massive immune suppression. So what we see is a difference between what we call battle stations. So we send all of our immune cells into this area to fight this infection, but if it happens for a long period of time, we start to get large amounts and massive amounts of immunosuppression.
Here, so that it could affect
Yeah.
these cells.
So all the sort of
So
like
what would be the advantage
inhibitions of
of
the
having
ways of
an
inhibiting the
open
immune system, like
endocrine system?
decreasing like MHC
Why
expression,
do
like
it
all
this
that? Are they
way rather than
all
this
referring to the inhibition of like?
They're talking about this chronic response. Yeah.
So in a condition where your body's in
Correct.
constant,
You need less concentration.
so are the glucocorticoids like trying to?
Because if all we're trying to do is to affect nearby cells, or the cell is
Often, yes. Yep.
the gastric pit.
Okay,
You
so
don't need
this is rats.
concentration because it's right there. You don't have to put it in
This is also true in people.
circulatory system, and it's going to
So, and this is one of my favorite experiments I've ever read about, in which they took a bunch of college students and gave them a disease,
specificity, right?
and gave them an illness.
You want to affect all cells in the
And I'm not
stomach.
kidding, they actually gave an illness to these college students. By the way, how can you do that ethically?
In the stomach. This-
Sign a waiver? No. I mean, everybody did sign a waiver, but everybody's required to sign the waiver.
Okay?
How do you run this experiment
Major
ethically?
advantages of
Give an
using
illness to
open
people.
endocrine system. Why is it called open, by the way?
Yeah?
Yeah?
Maybe. What illness are you talking about?
So because paracrine oftentimes is.
Yeah, I can't think of one either.
In the interstitial space.
Okay.
It's not actually out into the lumen folds.
Yeah?
Okay?
No,
So why
didn't
call it-
compensate them at all. Not in the least, not a penny.
Yeah?
Yeah?
Right, because it's actually secreting it outside.
Well, you could try, but then you're relying upon volunteers.
One way to think about it.
Everybody had to do this, and they did it
But
ethically.
in fact, that the digestive
Yeah?
tract
They already get sick and just use them at that point.
Oh,
outside of the body.
yep.
So in some way, the hormone
Vaccinations.
is treated in this open fashion.
Okay? So that's what a vaccination is, right? You're giving people an illness to train, at least a low-level one, to train their body in order to respond to it. So what they did is a very clever experiment. I loved it. Is that they got freshmen coming
How
in.
do we regulate?
Half of them were given their required inoculation in the first 2 or 3 weeks of the semester. And the others, half, were given their inoculation 1 week before finals. So guess who was more stressed?
Middle section of the stomach.
You guys aren't stressed before finals? Okay. So, and what they found was that the response to the inoculation was much, much higher earlier in the semester than later. So the high levels of glucocorticoids, just from that low level of chronic stress that all the students are facing as they're coming into their first round of finals, dramatically suppress the immune system, such that they did not particularly respond well to the inoculation. Kind of a cool experiment, huh? Okay, so why do this though? Why in the world would you inhibit an immune response as part of a stress response? This was so difficult for people to get their heads around that for about 40 or 50 years after people learned this, they had absolutely no idea what to make of this. It was in fact, papers used to talk about the paradox of glucocorticoids and immune function. About 20 years ago, a paper came out that suggested a reason why glucocorticoids might suppress the immune system. And it has to do with this. Okay, so you got a bunch of zebras. Now, one of the things that a very early researcher did when he was trying to understand the ecology and the biology of zebras out on the middle of the plains in the Serengeti is he's trying to study these. How do you know which one you're trying to study? They all look pretty similar. And so they're all sitting there mixing around in their herd. And so in order to tell them apart, he went out with a bunch of paint and paint markings on their flanks so that he could tell. So that was number one, that's number two, etc. What happened, though, was it turned out that all of his marked zebras got nailed by lions and other predators. Because what do you see? You see a whole bunch of herd. And how are you going to decide which one to attack? It becomes a little bit random. Not just random, but it's really hard to attack something that's constantly moving. It's just hard to focus on the individual animal. But as soon as they had a big mark on them, they immediately got targeted for predation. And so think about what would happen if you had a giant inflamed ear. Okay, so once you just got into a fight with another male and they rips apart your ear a little bit and it gets really, really red and juicy. I mean, what happens if any of you twist a knee? What happens? Blows up, right? Not only does it blow up with lots of inflammation, but it also starts to lock your knee so you can't run. If you're out in the middle of the wilderness with a lot of predators running around, that probably is a recipe for disaster. Not only have you just made yourself very noticeable to all the predators around, but you've also decreased your ability to escape. So maybe the best thing that glucocorticoids can do in that situation is inhibit inflammation and to decrease inflammation. So this is what people are now thinking about is the reason why glucocorticoids evolved to inhibit the immune system. Yeah.
Where acid stimulates the cell corpus, which secretes metastatic.
No, it's actually the opposite. It slows it down. Inflammation is actually really good for helping heal, fighting off infection. Yeah. Usually 2 or 3 days to shut that down. So the first thing it does is elevate the response, and then it starts to shut it down.
So the acid is sensing
Yep.
the acid. And then finally, in order to get...
So this was, again, this experiment here.
In the first place, we're also going to...
So you get this elevation in the immune response. So what people think, this is what I'm talking about in terms of, maybe I didn't explain this as well, the battle stations versus immunosuppression. So first thing that people did was when they looked at this, they'd inject glucocorticoids, and then they'd look at the amount of antibodies or B cells or T cells floating around in the blood. And immediately all of those cells decreased in the bloodstream. And it was these guys, Fergus Davar and Bruce McEwen, who argued in this paper. They said, well, of course they all went down. Let me come back to that analogy from a country that's just been attacked. If you've just been attacked, what's the first thing you do? You send out your army. Well, if you're sending out your army, that means they're no longer in the barracks, right? They're on the front lines fighting. So if what you did was went and looked in the barracks, you would say, oh my goodness, we suppressed this country's immune response after they got attacked because there aren't any soldiers left. Well, there aren't any soldiers in the barracks. They are on the front lines. So there aren't any immune cells in the bloodstream. Why not? Because they've left the bloodstream and are sitting there at the site of injury or infection actually fighting it. So the initial response is to mobilize that the immune response in order to get into those areas, and then and the consequence of that is seeing a decrease in your immune cells in the bloodstream. But if that keeps going on for a couple of days, then you start to see those cells not being replaced. Does that start to make sense?
Which stimulates the parietal cell, and you also get the direct effect of gastrin.
All right.
And you get the nervous system acting on the parietal cell. All of which results in the release of acid. We then get the negative feedback.
Okay, so that's the decrease, that's the effects on the immune system.
Which then shuts down acid.
We talked also about here, this decrease in inflammation.
Directly down the G cell.
We also talked a little bit about this decrease in metabolism, in the fact that during the gluconeogenesis, one of the things to do is decreasing the amount of glucose flat going into cells. Another thing is that glucocorticoids you can do is inhibit growth. So this is a really long-term effect, but you can see a long-term inhibition in growth and the decrease in growth hormone release when you have high amounts of glucocorticoids. And this is one of my favorite examples of this.
Okay.
You get this thing called stress-induced dwarfism.
We've got all our acid down.
Why am I putting up a picture of Peter Pan? Does anybody know the story? Yeah?
Now, the things that happen in the blood can't work with acid. So now we need to counteract the acid.
About a land of kids where they never grow up.
This is
Correct.
a hormone called.
It turns out to be very rare.
The secretin is.
It's the kind of thing where, child abuse doesn't normally do it, but locking a kid in a closet for 2 years, you'll start to see stress-induced dwarfism. It's got to be really severe, which is why it's quite rare. But it does shut down growth, and Barry is one of the people. We don't know this for fact. He was writing and grew up prior to us understanding glucocorticoids and this kind of response, so it couldn't be tested. But most physicians think that that's why he didn't grow to what he did. And was, again, this idea that he couldn't grow up, and therefore he wrote about kids.
So we have here now the exocrine.
All right.
Which is secreted.
So another thing glucocorticoids will do, they'll inhibit visceral activity. So again, they'll start to shut down the amount of activity that's taking place in the gut. This seems to be why we start to see increases in ulcers with stress. It's not that stress causes the ulcer. The ulcers we now know is caused by a bacterium that grows in the stomach. But one of the things glucocorticoids do is they start to shut down the proliferation of the cells that are lining the stomach. And so they make it more likely for H. pylori to cause an ulcer. Okay? Reproduction. Glucocorticoids are really, really good at shutting down reproduction. How do they do that?
And he was working at the
So they decrease gonadal steroid release. So they do that directly by decreasing GnRH release from the hypothalamus, they decrease LH release from the pituitary, and they decrease gonadal responsiveness to LH. So all of those things that we were just talking about, a couple months ago, glucocorticoids shut it all down. And they also reorient behavior away from reproduction. So if you are stressed, in humans, but also in animals, the libido goes down. Your desire to actually have sex decreases, the ability to interest in the reproductive behaviors. So not only are we shutting down the biology, we're also shutting down the behavior.
But one of the people in this lab then showed
So,
that, well, if you just put stuff
a couple of the really interesting things, though, that takes place is that there are some species in which glucocorticoids do not shut down reproduction. They've entirely removed this ability to shut down reproduction. And most of you are probably familiar with one of these species because it's very common, and that's in salmon. So this is a concept of seminal parity. So seminal parity means one breeding attempt and then die.
thereby showing there was a hormone. The interesting part about this story is that
So salmon,
it was so
I think most of us are familiar with the life history of a salmon. Not all salmon, some of them, not all species of salmon do this, but a lot of the Pacific salmon especially, and some of the Atlantic as well, they are born in the streams, they migrate down to the ocean, they'll spend a number of years getting bigger and bigger and bigger in the ocean, and then they will then migrate back up those rivers, spawn, and die. And the whole system starts again. So these are pictures that I took when I was in Seattle of a fish swimming up. You see dead fish near the places where they're spawning. And here's a fish in which its entire tail is in the process of losing its skin. The skin has literally come off of the tail of this fish. And if you look at more of the something from more of a textbook, this is the fish that's sitting in the ocean. This is one that is getting ready to migrate up the river, and this is one that's just spawned. And what you see is a massive failure of the entire body. The skin starts to slough off. They suffer from massive infections. The tissue starts to be degraded. Why does the tissue start to be degraded? Because it turns out that the thing that causes the death of these salmon is a massive increase in glucocorticoids. Now, what do glucocorticoids do? One of the things is break down protein. And so they're literally breaking down the protein of their bodies. And so there's no way for these fish to actually survive. If you do an experiment, you bring the salmon into captivity and remove their ability to release glucocorticoids, like remove parts of their adrenal glands, they will survive the breeding attempt. They will not die. And there are species, one of them is called steelhead, that actually go up, breed, and go back down to the ocean. And they'll do that multiple times in their life. So there's no particular reason that anybody knows for why it makes sense for the salmon to go up and spawn and die, but they do, and the mechanism is through massive amounts of glucocorticoids.
there's a
Yeah.
33 amino acid form that's released by the blood.
So the reproduction doesn't seem to be causing it. Nobody knows why that massive amount of glucocorticoids starts to increase. Nobody knows the mechanism for why that's happening.
And CCK
What we do know, though, is that the massive amounts of glucocorticoids are not shutting down GnRH release. They're not shutting down LH release. They are still, their gonads are doing fine.
just like secrete, acid gets
So they get up there, they can lay their eggs, they can use the sperm to fertilize. That's fine. So they are essentially shutting off that whole system from listening to the glucocorticoid signal.
CCK has 7 major effects.
Pretty cool, huh? All right, so that's fish. Who cares about fish?
Number one is that
Then you read about these guys.
Gallbladder,
This is an antechinus. It's a small marsupial mouse that lives in Australia.
bile acids.
And it turns out they are also semilparous. They will raise their pups,
Bile acids
they
are
will
released
nurse them,
from the gallbladder
just like a
into
marsupial
bile
will do. They will then have them breed, and then at the end of at the time of weaning, you start to see the entire destruction of the adults. They start to lose all their hair, and they die. The entire adult population dies after breeding. Yeah.
So earlier when we talked about starvation, you said that like
The
first
second
we
thing it
break down
does
our
is it
glycogen
works
and
at
then
the
fat
pancreas.
and then protein. So why are they not breaking down fat before protein?
They do, but they're not starving. You can give them these as much food as you want, and it doesn't help.
Digestive enzymes.
Because the glucocorticoid levels are getting this is the same mechanism as the salmon. Their glucocorticoid levels are so high that they're ripping apart their bodies. Doesn't matter whether they're getting
Third
enough
thing
food or
it
not.
does is it goes up
Yeah.
onto the pyloric sphincter and it slows
This is like just let the
gastric
pups are like
thinking.
kind of grown and then the adult population.
Yeah.
And this
Nobody
is
has a clue.
You've got
I mean, think about it. If that was so important to get rid of all of your adults,
start digesting it.
to allow the pups to grow into that space, why don't more species do it?
The stomach until we finish digesting what we
Right?
just got into the water. So it
Yeah.
slows down the
Males
gastric
and
emptying.
And females.
allows what's already
So males will do it shortly after after copulation. The females will do it after weaning.
The fourth thing it does is it
Yeah.
So
Increases.
break down protein to get more glucose into the blood to go to the muscles.
intestinal
Right.
blood flow.
Okay.
Yep. So what they're doing is, that's what I said, this is not an energy thing. These individuals are not starving. This is not starvation and starving to death. This is high levels of glucocorticoids short-circuiting everything. So they have fat. It's not like they don't have fat, they have it. But the glucocorticoid levels are so high that they're breaking down the proteins as well.
It regulates
Okay? Yeah.
So
food
are the
intake.
pups like left to grow up on their own
Yep.
and then just breed
Decrease
with
in
each other?
hunger.
Yep.
So it's after they breed their own kids that they.
Again, this would make great
Yeah, so
sense.
the pups, the newborns, the young of the year do not die. It's only the adults that have just finished breeding, and they breed once and they die.
The sixth thing it does is it acts permissively.
Yep, so they raise them till they're independent, and then they die. Yeah,
Are their glucocorticoid levels high while they're breeding their pups? Or while they're nursing them?
no, they start to increase right near the end of weaning.
It
Or
decreases.
right around weaning, the levels start to increase. And then they keep increasing, and they don't live another maybe 2-3 weeks until they die. Yeah, yeah.
If like they were to like miscarry,
Okay,
so they
so
got
these are the
pregnant,
major
but they
effects
didn't
of
actually have
CCK.
pups or live
It's
pups,
one of
but
the
they
most
still
important,
die right
and
after.
all of these
Like,
are the most important.
continue on to another
Start
cycle.
to truly
And maybe have a
orchestrate
successful
all
breeding
of the
next
effects.
Great question.
that take place during digestion.
Turns out they'll die anyhow. It really seems to be something programmed. You get this massive increases in glucocorticoids, and then they're
Some of the
dead.
students came to my office.
Yeah.
If they don't
before
breed,
the
do
exam,
they die? Like, do they?
diagram
Yep.
what CCK.
Yeah, you can't keep them alive. Well, you can, you can keep them alive, but you do it by removing their adrenal glands so that they can't see this massive increase in glucocorticoids. If you do that, you can get them to survive and breed the next year.
Breeding,
So it really is this massive increases in glucocorticoids that's just killing all of these animals.
acid, and fats.
So, as I think I've told you before, 2 years ago I spent my sabbatical in Australia. My big goal for my entire sabbatical was to take a blood sample from an antechinus. I never got to do it. There was nobody around me who was actually working on it at the time. So a lot of this work was, the really interesting work was done back in the 70s and 80s on these. But really cool. This is a mammal that is semilparous. programmed death. Not programmed cell death, programmed organism death. And like I said, the ecologists haven't shown a whole lot of interest in trying to solve this problem, why this is taking place. But really, we still don't have any idea. I mean, one of my favorite hypotheses for salmon doing it was that their dead and decaying bodies were going to be fertilizing the area where their young were hatching out with a new fry. That has been completely debunked. That is not, that all of those nutrients are flowing downstream. They're not available for the growing fry. So it's not like they're fertilizing all this new eggs. You know, are you leaving space for your young to move into so that you're not decreasing the competition with your own young? That's another favorite of people. But again, it doesn't make any sense, because if that was true, why don't we have a kid and die? You know, why don't lots of species? You know, this is quite rare to see species in which they have this programmed death. So we think that it probably has to be useful in some way, or else it would be so potent to, if you could survive to the second year and get a second brood out, you would have such an enormous advantage over all of the other individuals in your species that you'd think that evolution would immediately drive it so that seminal parity disappeared. So there must be a good reason, but nobody knows what it is yet. Yeah. Yeah, there's about, as far as we know, not every marsupial does this, and it's mostly the marsupial mice. But there's also these things called quolls, which are about this large, that also do it. I haven't counted them up, but somewhere between a half a dozen and a dozen species have been shown to do this.
I mean, like species themselves. I mean, like
We're going to
strains of species,
slow.
like geographically separated by any other factor.
empty.
Do we know if all seven factors, like marsupial mice, are all subspecies of the mice?
And CCK also is going
Yeah.
to.
Yeah, it's baffling, isn't it?
increase
And the mechanism is glucocorticoids.
blood flow.
So you
Yep.
said if you remove the adrenal gland, they can still reproduce multiple cycles then?
Yep.
And they tried
Cover
that
this in
with
a
the
minute,
salmon to see
but
if they would
another
come back
hormone,
afterward and still not
VIP,
completely deteriorate?
So people haven't, I don't know if people have done it. It'd be much, much more difficult in salmon. Partially because they have lots of tissue that's there for glucocorticoids. The fish don't have a distinct adrenal gland, and a lot of the tissue is actually embedded in the wall of the kidney. So in order to really do, like a mammal, you can just take out the adrenal gland. For a fish, you'd actually have to take out most of the kidney. And that, as you can imagine, is not possible to do. That would be lethal. But what's interesting is that nature did that experiment for us. So a very closely related species is the steelhead trout. They are also salmonids. They're in the same genus. And so they do not show this massive increase in glucocorticoids, and they'll go up the streams, breed, and go back down to the ocean. So they'll do it multiple times. Yeah.
They both have a similar like the advantage of
the
breaking down
active
your
portion of the
protein
body,
and fat storage.
receptors is in that amino acids.
It's not. The first thing you want to do is break down your fat. This is sort of what people think of as it's a last-ditch effort. So you've used up all your glucose, you've used up all your fats. The only thing left is either to die or break down protein. So let's break down some protein first. So it's that last-ditch effort to survive.
So there's
Why
two
does
different forms.
the stress response itself skip that fat breakdown?
The stress response doesn't. Glucocorticoids do. Okay, so the epinephrine is really involved in breaking down both your glucose, right, to get rid of that glycogen, and it also has a major effect on inhibiting insulin and the production of fat. So don't think about it. Remember, there's two different aspects of stress. There's the catecholamines, the epinephrine, and then there's the glucocorticoids. Yeah.
You know if anybody is like
Anything else?
taking out the adrenal glands before that
Okay.
maybe that glucocorticoids
Just really
is,
quickly,
activating,
we can finish off digestion.
and
The
most-
hormones involved in digestion. So the CCK being released by the duodenum. The gastrin.
Yeah, so at least in these antikinase,
and right-
the surge in glucocorticoids takes place after breeding. So
Gastrin-inhibiting peptide.
it's really what happens after breeding. That's when you need to
We're not even sure that
adrenalectomize.
the GIP
Yeah.
is being released from the small intestine, but we're not sure there's much of a biological activity. The other one is mortillin.
Oh, yeah.
Mortillin is released down at the
I don't know
end
this for a fact.
near the
Uh,
small
n- n-
intestine.
I know for a fact.
Now you're
I don't know
starting
a
to get
person,
all this chyme coming
because
down.
I've never gone
Mortillin
fishing in
is released,
Alaska.
and it creates what's
But
called
I am told that if you're not at the river mouth, don't bother. The only thing that likes migrating salmon once they enter the river mouth are bears and eagles, because it apparently is absolutely disgusting. 'Cause what you're talking about is a bunch of flesh that's in the process of melting away.
Think about it squeezing and squeezing and squeezing.
Yeah, it's
and
really-
squeezing and squeezing.
Yeah, people say don't even bother. That just becomes sport fishing then. You just grab them and you release them because it's not worth eating.
75 to 90-minute
Okay,
release.
something to think about.
You can get that slow moving of the digestion,
All right,
stuff down through the tract.
running out of time here, so let me skip a few things and just talk about maternal effects. This is really the area of stress research that is probably most studied today. It's probably a lot of the most exciting for researchers. And that is that you get this transfer of glucocorticoids to the offspring. And it can happen through the placenta. It can also happen through in breast milk.
Intestinal.
And what it does is it alters the stress response of offspring as adults.
Released mostly by
And the way it does this is kind of cool. So what it does is it decreases or changes. So a moderate amount of stress,
is involved in increasing blood
so
flow
moderate stress
to the digestive system.
increases the glucocorticoid receptor
Okay, so those are the major hormones.
in the hippocampus.
Any last questions?
Whereas
Okay. Let's see everybody.
high stress will decrease GR in the hippocampus. So what does that mean? So think about it in terms of the decrease in the GR in the hippocampus. The hippocampus, if you remember, is a major site for glucocorticoid negative feedback. So if you lack the GR in the hippocampus, your negative feedback is decreased. So what you see is this is the stress response of a normal individual. This will be the stress response of a individual whose mother was suffering from stress. So you have lower amounts of GR in the hippocampus, and so therefore lower negative feedback, and so you see a much longer response. And people are showing this now in humans as well. There's a massive change in the way in which people are reacting to stress. And it can take place very early. So you stress the mom, what you end up with is a stressed offspring. And the things that have gotten people really worried is at least these rodent studies where you can show that if you stress mom, you get a stressed daughter who then produces a stressed granddaughter, who then produces a stressed great-granddaughter. And so just a huge amount of stress can then percolate through the generations. And so people are starting to see that this may be one of the reasons why low socioeconomic status in humans ends up percolating through generations, because you end up changing the actual way in which the brain is being built, especially in places like the hippocampus, because of the stress that your mother experienced. Yeah.
I have watched a documentary where they actually were able to test people with this because a bunch of moms got stuck in a huge snowstorm in like the early 1990s, and they couldn't get out, and they weren't sure if they were gonna have enough food because it was a huge ice storm. I think it was in Canada, and they followed the kids for years and saw that they had decreased intellect, they had a bunch of other health issues, they had to have all these different things. It was all because the moms were so stressed out when they were pregnant, and they had no idea when they were gonna get food and get out or get warm because they're running out of all these supplies and stuff, and they
Yep.
really
There's a
saw
lot of
the
studies,
causes.
people are looking at this. It's also true that don't blame your mom, at least don't blame your mom exclusively, because a paper came out last year, here you go, that's the sperm. Turns out that this really baffled people. But you can show through epidemiology studies, just by looking at people, that a stressed dad also can change the way in which the offspring react. Nobody had a clue how that was taking place because there isn't any DNA, the kinds of DNA that's part of the same thing with mom. But it looks like the epigenetics